Hi everyone, and welcome to our last lesson of the entire semester. If you're here, great job. You guys are amazing. I know it's been such a weird semester, and I hope these videos have been helpful for you along the way. But just great job. You guys are awesome. So today we're going to talk about mitosis. This is our last little piece that we're going to learn about in biology. So first we're going to refresh what we did last week, and then we'll learn what mitosis actually is. So grab the note that goes with this and we'll get started. Okay, so let's refresh first. So last week we talked about the cell cycle and it looked kind of like this. We have interphase, which is this really long section here that's split into three parts. Remember, we have G1, which is our growth phase. And then we have our S phase, where our genetic material is making a copy of itself. So all of our chromosomes make a copy of themselves. Then we have a second growth phase. And then this is the part we didn't talk about last week. This is the mitosis part here. And you'll see that I've broken it up even further. So all of a sudden we have four sections in this mitosis section here. And so those are prophase, metaphase, anaphase, and telophase. And mitosis is where cell division is actually happening. We actually have one cell that is splitting apart into two new daughter cells. So again, those are our four phases: prophase, metaphase, anaphase, and telophase. And the way that I like to remember this is with a disobedient dog. PMAT. That's how I like to remember it. So if you're ever trying to remember the order of these phases of mitosis, you can just picture this dog's face and hopefully it will come back to you. Okay, so now what are these phases? What's actually happening here? Before we can get into that, we're going to look at some new terms that we haven't talked about so far. So let's look at some new words. Now you'll notice, I just want to pause quickly, you'll notice in biology so far that it's a little bit different from chemistry in that we don't have a bunch of practice questions or rules and exceptions and things like that to try. Biology is much more terminology based. It's much more trying to draw things out and see how they work. So instead of doing practice questions, I do suggest that you look over each of these notes that we've done so far a few times until those words really start to make sense, until the picture in your mind is there. So that's something that I just wanted to say is a little bit different from our last unit. Okay, so let's see these new terms. So first of all, we're going to talk about chromatin, and those are long strands of DNA located in the nucleus. And you might be like, what? I thought those were called chromosomes. Well, this is just a different form that they take. And this is actually the form that they're in most of the time. And I call it spaghetti form. So you can see in this picture that the DNA looks like spaghetti. It kind of just looks like random long noodles all over the place. And so that is called chromatin. And this is when the DNA is really spread out and it's just long and it's noodly. So that is how it likes to exist most of the time. When it's getting ready for mitosis, that's when they shrink, they kind of compress into those X shapes, and that's what we call chromosomes. So it's the same thing, it's the same genetic information, but just a different form that it takes. Yeah, so here, when we, all of a sudden those spaghettis, they compress into this X shape, and this is a chromosome. And something that's important to mention is that this side of the chromosome here, I don't know if you can see my mouth, hopefully, this side of the chromosome is identical to this side of the chromosome. So we've got a copy here. We've already gone through the S phase, and this side has made a copy of itself right here, okay? And so each copy is called a chromatid. So this is one chromatid, and this is the exact copy of that chromatid. And together they're joined in the center, and the whole thing is called a chromosome. So that's a lot of different words that are very similar to each other. So chromatin, chromosomes, chromatid. It's all talking about the same genetic information. It's just the form that it's in. So if we're in spaghetti, it's a chromatin. If we're in an X shape, then the whole X shape is called a chromosome. And within that X shape, we have two identical copies of those genes, and each copy is called a chromatid. So the middle of the X there, how those sister chromatids are attached is called the centromere. And I always think centromere, center. It's the center of the chromosome. So it's what's holding those two things together. And it's also where something called spindle fibers, which we're about to talk about, that's where they attach, is this little part here. Okay, centrioles. So these are organelles that we haven't talked about yet. So we've talked about ribosomes and mitochondria and a few others, but we haven't talked about centrioles. Centrioles are these tiny organelles that like to exist in pairs, and they produce something called spindle fibers. And spindle fibers are web-like strands that attach to the centromeres. So I like to call centrioles Spider-Man, and they like to shoot out these web-like strands to attach on to the centromeres here. All right. Now let's talk about mitosis. So cell division in general happens in two parts. We've got the separation of sister chromatids. So we have the actual genetic material being separated into two new cells. And that is what mitosis is. And then we also have the splitting of the cell itself and all its organelles. As we talked about with the cell cycle, the cell is going to be growing bigger. We've got two growth phases where the cell is growing bigger and it's producing a lot more organelles. And that's so that when we split up into two new cells, those new cells have enough cytoplasm and have enough organelles to be able to survive. So we've got two things happening in cell division, and they kind of happen at the same time, but we're kind of going to split it up as we talk about them. So for mitosis, like we said with PMAT, prophase comes first. In prophase, the chromatin, so the spaghetti form, is going to thicken and condense into visible chromosomes. The membrane around the nucleus is going to start to dissolve, and it looks like this. So we go from spaghetti form here to visible chromosomes, and this nucleus that is containing the genetic material starts to dissolve. So that's going to start to go away. And you'll notice these little guys here, those are our centrioles. So those are the organelles, those are our Spider-Mans. So you can see also that they've migrated to either end of our cell here, and they're starting to produce spindle fibers. Next is metaphase. So in metaphase, there's no more nuclear membrane, it's completely dissolved, and chromosomes are lining up in the center of the cell. And this is where the spindle fibers will attach to the centromeres. So something that I like to remember this stage by is M for metaphase, M for middle. The chromosomes are lined up in the middle of the cell, so I kind of think about it that way. And then we can see that our Spider-Mans here, our centrioles, have produced spindle fibers that are long enough to attach to the centromere of each chromosome. So we're about to split apart. If you think about these X's, remember that the one side is the exact same as the other side. So we're about to pull those apart so that each new cell can have a perfect copy of all the genes. And that is exactly what happens in anaphase. Think A for apart. So our sister chromatids are separated, they're being pulled apart, and they're going to be pulled into the centrioles at opposite ends of the cell. So just like this. So this is kind of neat. Our Spider-Mans, they throw out these webs, they attach to the centromeres of our chromosomes, and then they kind of pull it back like a fishing line. They kind of reel those spindle fibers back in, and what that does is that pulls really hard on the chromosomes until they eventually split. So here's one half and here's the other half. So they've been split apart, but you'll notice that each side here has the exact same amount of each chromosome. So that was confusing. Let me elaborate on that. We've got 1, 2, 3 red, 1, 2, 3 red, 1, 2, 3 blue on this side, and 1, 2, 3 blue on this side. So what that means is that both of these new cells that are going to be forming have exactly a full set of all of the chromosomes. All right, telophase. So this is where we really start to see daughter chromosomes and daughter cells forming. So those daughter chromosomes are going to stretch out to become chromatin. They're going to become spaghetti form again. We're going to have 2 new nuclear membranes forming. So we're creating the nucleus in both of our daughter cells. And that kind of looks like this. So we've split up. These are going to stretch out and turn back into spaghetti form. And we've got these nuclear membranes that are starting to form around the chromosomes. So then we talked about this idea that we've got the separation of the chromosomes is part of mitosis, and then the second part was the separation of the cytoplasm and all the organelles. And so really that's what this is. It's called cytokinesis. And cyto meaning cytoplasm, kinesis meaning separation. So this word literally means the separation of the cytoplasm and the organelles that are in the cytoplasm. So we kind of consider this part of telophase. They're happening pretty much at the same time, but for our purposes, you can think of them as 2 different phases. So we've got the cytoplasm and the organelles that are being divided into the 2 new daughter cells. And then eventually the cell membrane is going to pinch in the middle to finally, kind of like a balloon, you can think about if you twist a balloon in half, it's pinching in the middle there to form 2 completely new cells. And we can see that our chromosomes are back in spaghetti mode. So here's the whole thing. Here's PMAT. As a whole, we can kind of see that we have no more, our nucleus is dissolving there. We've got spindle fibers starting to form. They line up in the middle, they separate, they go apart, and then we form 2 new daughter cells. So let's watch another ridiculous song to help us remember this.
Guys, I want everyone's attention. We're going to go over what we did yesterday. We're going to go over the stages of mitosis. All right, everybody ready? All right, let's go. Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly, but note the nucleolus, the ribosome factory outside the nucleus, the 2 centrosomes. They later make a spindle which will pull apart the chromosomes. Prophase follows, the chromosomes condense, each is made of 2 sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears and melts away as the cell takes a ribosome production holiday. The centrosome separates, starts spindle formation, the separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from 1 cell to 2. Mitosis, how cells renew. In late prophase, pro-metaphase, the nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle wend and wind. The spindle is made of microtubule fibers which attach to chromosomes. A kinetic causes a protein patch that serves like a handle that the fibers can grasp. When they pull apart the chromosomes, splitting them in half, the spindle moves the chromosomes with nudges so fine into linear formation on the 50-yard line, a location equatorial, defined in metaphase, where the chromosomes are lined up in that middle place. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from 1 cell to 2. Mitosis, how cells renew. The spindle fibers pull on the kinetic cores. A cellular, molecular, mitotic tug-of-war. The centromere snaps, sisters get separated. Now these chromatids are chromosomes. They've been upgraded. The snap and separation defines anaphase. The A for apartness, for moving different ways. Kinetic core, spindle fibers separate. The sisters see I'm waving goodbye, calling out I'm gonna miss you. And the other spindle fibers pushing grapple like felons, make the cell elliptical like a watermelon. In telophase, membranes form around the chromosomes that spread out as a nucleoli come on home. Mitosis, chromosomal ride in a pro-meta-anaptelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In animal cells, there's a ring of microfilaments that form at the equator, and they cinch themselves in tighter till the cell is in two pieces. Yeah, in animals, that's cytokinesis, but it's different in plants, and then the cell divides by building a new cell wall from the inside as the Golgi sends vesicles with cellulose goo, which makes a plate, then a wall divides the cell in two. And instead of one mother cell, we now have daughters too. Identical twins, kind of old but kind of new. From your single cell beginning, this is how you grew. And for single celled eukaryotes, it's reproductive too. Mitosis, chromosomal ride in a pro-meta-anaptelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Mitosis, chromosomal ride in a pro.
All right, that guy's great. So one thing I did want to point out is he mentioned centrosomes. That is another word for centrioles. So just don't be confused there. Our Spider-Mans, we're going to call them centrioles, but they can be called centrosomes too. Okay, so now let's just do a quick quiz. You can use this to just see what you've absorbed in this time that we've been talking. So I'm going to show you a picture. You can pause the video and see if you can guess based on the picture which phase that it is in. To really get the most benefit from this, I would suggest not using the note. But yeah, let's just go through and see what we know. So take a look at this one. All right, this is prophase. Hopefully we can tell by the dissolving nucleus here, by the fact that our chromosomes are visible, they're condensed, and our Spider-Man, our centrioles, have moved to both ends of our cell. Try this one. This is cytokinesis. So we have our, first of all, notice that we have two cells instead of one, so we know we're near the end of mitosis, and our DNA is back in spaghetti mode, and our centrioles are no longer producing spindle fibers. And the real defining thing of cytokinesis is that we've actually had a separation of the cytoplasm here. Next up, this is anaphase. Think anaphase, A for apart. You can see that these have just, all these chromosomes have just been pulled apart, and they're being reeled in by the centrioles at either end to be able to create two new cells. This one is interphase. Kind of a little bit tricky there because that wasn't in the PMAT that we talked about, but something that we can tell here is that we've got only one cell. So we know we're at the beginning of mitosis. We've got spaghetti form, so we're not quite in prophase yet. So, and our centrioles haven't moved. They haven't moved to opposite ends. So this is before prophase, and what comes before prophase is interphase. So this is interphase here. This one is metaphase. M for middle. You can see the lineup of chromosomes in the middle of the cell. So this is metaphase. And at this point, our spindle fibers are attaching to the centromeres of our chromosomes. And last one, this is telophase. So we've got two, first of all, we almost have two new cells, so we know we're near the end of mitosis. We've got new nucleuses forming in both of our cells, and we only have half. We only have the sister chromatid in each, so they've been pulled apart already. So that is telophase. And that brings us to the end. So I hope that you found that helpful and informative, and if you have any questions, as always, please feel free to email me. Have a wonderful day.